It's a silent killer, also described as a hidden health epidemic. Fatty liver disease. The early signs are subtle, but if left untreated, they can cause a variety of illnesses, including diabetes, cardiovascular disease, and even some types of dementia. There is no cure, no treatment, other than changing to a healthier lifestyle, but there is hope. Fatty liver disease can be deadly. Around a quarter of all people suffer from some form of this disease, and there's a critical shortage of donor transplants. But a biotech company in Oxford is hoping to change this by developing a genomic medicine that can switch off fat-causing genes. Wayne Eskridge is not an unusual case. Poor diet and exercise saw his weight increase over the years.
My doctor wasn't concerned. I wasn't concerned. I didn't realize the risk that I was taking.
What he didn't know was his liver was also building up dangerous levels of fat.
The thing about the liver is that it's so tough. It's very forgiving. You can just beat the heck out of it, and it just keeps working. I was incidentally identified during an operation in 2010 for a gallbladder removal. And the surgeon at that time came out and told my wife that I had cirrhosis and I had maybe a couple of years to live. And this takes us into all the problems that people have with this disease, because none of my blood tests showed that.
Eventually, Wayne asked to see a liver specialist and discovered he had stage 4 cirrhosis that causes severe scarring and poor function, often seen at the terminal stages of chronic liver disease. His experience led him to set up the Fatty Liver Foundation in the US, where it's thought around 100 million have some form of fatty liver disease.
I was very fortunate that at the last minute, my doctor and my wife pulled me back, and we changed our lifestyle.
In England, at Oxford University in the Innovation Lab building, Okabio co-founders Dr. Quinn Wills and Jack O'Meara have developed a therapy they hope could successfully treat fatty liver disease and be delivered straight into the liver.
The biggest problem by far really is that a lot of the problems we're looking at, like fatty liver disease, are silent killers. They're silent for 10, maybe 20 years before you present with complications, and then sometimes the therapies are just too little too late.
Because the disease is so silent, the team couldn't figure out how to measure changes in trial patients. They soon realised that working in the transplant space could solve a lot of problems.
So let me take you on the final part of this journey. If you're squeamish, look away now.
Yeah. So this is a fatty liver?
This is a fatty liver.
A perfusion machine can keep livers alive before transplant for up to 24 hours. At Birmingham Hospital, donated livers not healthy enough for transplant are put on this machine so they can be tested with Okabio's therapy.
All they need to do is inject the therapy into the blood. It circulates, goes into the liver cells, and by the time the liver is transplanted into the human, it's in the liver and stays in the liver for months at a time.
The therapy contains instructions to switch off or turn down the genes creating fat. In future, they hope to be able to inject it straight into a patient. And so it's not cleaning it at all, it's just sort of making the fat less harmful.
That's exactly it. Boosting the metabolism so that over the months the liver is better at clearing out the fat, but it's also better at responding to the stresses that the fat might cause.
Wow. And just one dose?
One dose. Single medicine lasts for up to 6 months at a time.
Incredible. Even though this genomic technology is still very much in the testing phase, consultant surgeon Dr. Heineck Mergenthaler says it is promising. So if the therapy does prove reliable and successful, how could that change transplant medicine?
If we were able to treat the fattiness of the liver disease, that would be a game changer for having more transplantation, more donors available. But in general, transplantation is only a tip of iceberg, because if we can treat the fatty liver disease on the machine or when outside the donor, we will be definitely able to treat the liver inside the human afterwards. So the need for transplantation would really decrease.
But right now, the need for donor livers is desperate.
we are starting accepting livers and donors, which we wouldn't consider 10 years ago, because the liver disease has been increasing. There has been about a fourfold increase in liver disease over the couple last decades. So the need for transplantation is really booming. Unfortunately, 1 out of 7 patients currently dies on the waiting list because there is not available donor for them.
The liver is the largest solid organ in the body and is vital to the metabolic system as well as detoxification and the immune system functions. It also controls the body's ability to break down fats. While alcohol can affect a person's liver function, it's not totally to blame. Non-alcoholic fatty liver disease is the term used for a range of conditions caused by a buildup of fat in the liver and is usually seen in people who are overweight or obese, but not always. It's also the main reason for needing a transplant in countries such as the UK and US, and for the shortage of healthy donor livers. In medical science, biomarkers can be used as an indicator of disease. For example, blood pressure is a biomarker used to determine the risk of stroke. But when it comes to fatty liver disease, there's a lack of them, so researchers don't know if the treatment is working.
Either you go into the lab and find biomarkers, or you find a way to do trials without the biomarkers. And the latter is really the solution we've gone for as a company.
Instead, they're figuring out which genes cause dangerous fat and testing which has the greatest influence on disease development. So Quinn, are you reprogramming livers?
Yes, it's a very exciting time these days, to completely change how we think about medicine. Traditional medicines really target proteins using small molecules. These days, many of us are directly targeting genes in the DNA and how they talk to the rest of the body to make really big changes for long periods of time.
So how do you stop genes making fat? At the centre of a cell is the nucleus. Inside are thread-like structures called chromosomes. Each chromosome is made of a single molecule of coiled DNA that carries the genetic blueprint of the proteins that the cell needs to make. Whenever the blueprint is read, messages are sent from the nucleus to the cell, telling it which proteins to make and how much of each. These messages are sent using another molecule called RNA. It's some of these messages that Dr. Will's team is trying to block. They do this by making a synthetic, long-lasting mirror version of the specific RNA message they're trying to block. They also add a signal on the end of the mirror message, so when it's floating around in the blood, it can find and attach to liver cells. The liver cells take in the synthetic mirror message, which binds to the natural message, causing it to break down. By doing this, they are dialing down specific proteins that are in excess when the liver is full of fat. This type of medicine is called RNAi, or RNA interference.
It's a big breakthrough, not just at Oka Bio, but in deep phenotyping in general. The ability now to be able to study genomics at the level of cells, which we couldn't do 6-7 years ago, is equivalent to when the physicists began smashing atoms to see what's inside.
Traditional genetics connects DNA with disease, but Dr. Wills and his team argue that it doesn't answer important questions about chronic disease, such as what the gene is doing, at what stage in the disease it plays a role, and in which organ. Their technique, known as deep phenotyping, does just that. By imaging and sequencing organs at incredible detail from healthy individuals all the way to very ill patients, they build models of how changes in our genes, cells and organs over time eventually result in disease.
We start off with liver biopsies, and we're working together with transplant surgeons here in Oxford to image and genotype and sort of analyze the genes for thousands and thousands of samples. Here's a good example of how we do it. You can see 2 liver biopsies here. And if you look very carefully, you can even see regions where the liver is beginning to break down. So we use imaging AI to really scan all of these images and identify thousands of these types of features that we think are important. So here, imaging AI has identified in these blue regions areas that we call ballooning degeneration, which we believe is important in this disease. It's these cells in particular that we're trying to understand because we believe they are the cells that connect fatty disease with eventually fibrosis and cirrhosis.
How do you figure out which genes are actually creating this fat?
By using these methods, we build up a very detailed computer model of what we believe the liver is doing and which genes are going up or down or changing the behavior in response to or driving the fat changes in the liver.
So you're feeding data into the computer, and it's the computer that's telling you which genes you need to hone in on.
Absolutely, right down to individual genes and individual sequences of genes if we need to.
But it's up to the scientists to test human cells to see how they respond to different levels of fat.
This is Kenny. What he does is he's testing out the ideas that we got from the computer and the predictions in little micro livers that we culture in those trays.
I mean, you say micro livers, I'm expecting to see, lots of it, but these are actually sort of what cells, liver cells?
Liver cells. If you look a little bit closer over here, you can see what they look like with fat. We're showing the fat in green. This is a close-up on a micro liver where the cells are building up fat. We've shut down one of the genes on the left to build up more fat, to understand what happens when fat builds up. And on the right, we've shut down a gene that we think might make for a good therapy because it turns down the level of fat that the cells are accumulating.
50% of obese people who have liver transplants because of fatty liver disease redevelop it within a year. Dr. Wills wants to prevent that by giving the therapy every 6 months and ultimately giving it to those living with a fatty liver that have yet to develop complications. He also wants to dial genes up as well as down.
There are genes that really speed up how fat is metabolized in your cell. And those ones we really do want to augment. It's just a much tougher problem than switching ones off at the moment.
Okabio is hoping their research will not only save lives, it will give the doctors and surgeons greater insights so they can tailor the medicines to the patient.
We're building this 1,000 liver atlas. It'll be the most detailed genomic atlas of the liver that's been developed to date to really study what's happening in the liver at a genetic level.
This is a first step towards really mapping out the body at a cellular level and how cells not just function normally, but how they start functioning as you start developing too much fat, as you become obese, as your cholesterol levels go up, as your blood pressure changes, and how those in some individuals might improve your risk or worsen your risk in terms of getting a disease.
The Atlas is a collaboration with Oxford University, and the data will be publicly released at the end of 2022. Not long after, they hope for the first time to be able to treat a transplant liver just before it goes into a patient. Wayne Eskridge saved his life through diet and exercise, but for many that won't be enough.
In the US, there's 100 million Americans who have fatty liver. Today, it is steadily moving into the younger generations. We're getting it earlier and earlier in life. We badly need effective drug therapy. It's going to be devastating to medicine and, not just medicine. I mean, it's devastating to society and families and people because chronic illness is a nasty way to die.